Hey class, in this video we will be focusing on the structures of the spinal cord. We'll save the spinal nerve for another video. Just as a quick reminder, in this class we're trying to use current A&P terminology, and we as human beings are bipedal organisms. We are not quadrupedal organisms. So what does that mean? That means that dorsal and ventral don't really apply very well to human beings. Anterior and posterior are better terms to describe the directional terminology on a human being. With that said, dorsal and ventral are pretty heavily embedded in the literature, so you'll still see it quite frequently. So let's dive into it and talk about the gross structure of the human body. I'm here at the system view in Anatomy Atlas, and I'm just going to click on nervous system. So here's our default view of the nervous system. So here's a human body with a nervous system and a skeleton for reference. I'm going to go ahead and remove the ligaments and connective tissues there. And we have our nervous system. I'm also going to go ahead and remove the skeletal system as well. So now we have just the nervous system present. So as we're looking at this nervous system, we can see that it's, well, kind of complicated. We have spinal nerves that are shown in yellow, and then the central nervous system here is pretty much going to be surrounded by this pink membrane, which represents the Dura Mater of the brain and Dura Mater of the spinal cord. And since we're focusing on the spinal cord in this video, I need to remove those nerves of the peripheral nervous system. So I just clicked on a random one. I'm going up here to the breadcrumbs, and I'll scroll up to where I can just highlight the entire peripheral nervous system, and then I'll hide the whole peripheral nervous system all at once. So here's our cleaned up view of the central nervous system. So we have first some gross structure to the spinal cord. We have the cervical enlargement. We can see that cervical enlargement. Oops, let me hide the Dura Mater there. So there I just hid the Dura Mater. The cervical enlargement is visible, easily visible here with invisible body, as well as this thicker portion of the spinal cord that's right here in the middle of my screen. If I click on it, visible body doesn't allow me to have this as a sub-region, but we know just from the morphology of this structure that this region of the spinal cord is referred to as the cervical enlargement. The next one that we're going to cover is the lumbar enlargement. Now, as we look at our lumbar enlargement, that lumbar enlargement is going to be down by the lumbar region, and it's not as well pronounced as the cervical enlargement. So if I zoom out, the cervical enlargement is still very clearly visible. But to see the lumbar enlargement, I'm going to need to zoom in just a little bit more. And it's a more nuanced enlargement. Since it's more nuanced, I'm going to go ahead and annotate here so it's a little bit easier for us to see. Our lumbar enlargement starts approximately right here and goes to approximately right there. So for this region of the spinal cord, it's a little bit thicker. And this thickening of the spinal cord corresponds to the lumbar plexus and the sacral plexus, a bunch of spinal nerves that are going to originate and insert here at this region of the spinal cord. So it needs to be a little bit thicker. Next we have the Conus Medullaris. Sometimes in other texts you'll see this referred to as the Medullary Cone. The Conus Medullaris is going to be visible down here at the apex of the spinal cord. It comes to a fine point. This fine point is called the Conus Medullaris. And there's also going to be a bunch of filaments that extend outward. These filaments, I'll go ahead and zoom in on them a little bit. There, we've centered them, are referred to as the cauda equina. And the reason these are referred to as the cauda equina is because it kind of resembles a horse's tail, especially if you look at it from a lateral view like this. You can see the curvature of it. So when we see this term cauda equina, think horse, think tail. These filaments are going to be located within the lumbar vertebrae. So I'll go ahead and add the skeletal system back so we can see, hey, there's those lumbar vertebrae that go around the cauda equina. Something that's confusing for a lot of students is when we look at the lumbar region the lumbar enlargement right here on the spinal cord, that lumbar enlargement is actually going to be surrounded by thoracic vertebrae. So that's kind of counterintuitive. Next we have the filum terminale, also referred to as the terminal filament. In some probably high school level, I would say A&P textbooks. And as the name implies, as we look at these filaments, we're going to have one filament go all the way to the very end of the vertebral cavity. This is the filum terminale, and it serves as an anchoring point to help anchor the spinal cord in position. To add our skeleton, you can see the filum terminale goes all the way down. I'll hide the coccyx. Goes all the way down to the coccyx. So I'll add the coccyx back just so we can see that again. Let's click undo there. Try that again. There we go. So the filum terminale goes all the way down to the coccyx bone. So that's our overview of the gross structure of the spinal cord. Let's look at a cross-sectional view of the spinal cord. We can access cross-sectional views in visible body a couple different ways. One of the ways is that we can just click on the spinal cord. Let's get rid of that skeleton again. The keyboard controls were changed recently, so now I'm a little yutsy-futsy with visible body. So I'll go ahead and hide this section of the spinal cord, and then we can just zoom in and look at that cross-section. But this is a cross-section that's devoid really of the greater context within the spinal cord. So I'm going to back up here. To give you a little bit more context, I can click on cross-sections in visible body and then access cross-sections of the human body. And I want to go with a transverse slice. We'll do a transverse slice kind of right in the middle of the thoracic cavity. So we'll say T5 to T8. So now we can see the cross-sectional slice of the spinal cord right here with the greater context of the vertebrae and all of the other organs of the body. Something that's cool about this program is that we can also see the equivalent section as a cross-sectional slice in a human cadaver. So we have our spinal cord right here highlighted at the tip of my mouse. That same structure is now at the tip of my mouse on the left-hand side of the screen. And I can click MRI scan, and you can see the same equivalent structure right here within an MRI scan. So this is we have three different ways of viewing it. We have an MRI, we have a cadaver, and then we have the visible body cross-section. We're going to spend most of our time focusing on the visible body cross-section. So when we look at this, I first want to emphasize that we have anterior, posterior. So I'll go ahead and annotate. We'll say, actually, let's pause for a moment here and just center it in our view. There we go. So to make sure we have our correct frame of reference, I'll put an A on the top of my screen for anterior and a P down here for posterior. So now we have our frame of reference. So first, let's talk about the gray matter. As we look at gray matter is going to be, well, gray, as the name implies. So when we're looking at this, we have this gray substance in the spinal cord known as gray matter. This is going to have mostly the neurosoma of the neurons. So we don't have a lot of myelin. Instead, we have the unmyelinated parts. Towards the anterior of the spinal cord, we have the anterior horn. I'll go ahead and highlight that right now with green. There's our anterior horn of the gray matter. In red, I'll cover our posterior horns right here. And then in blue, I'll do the lateral horns. So we have lateral horns of gray matter as well. In the very center, in black, we have the central canal of our spinal cord. And then wrapped around the central canal, we have the gray commissure. And I'm going to use some yellow to emphasize this region. You can think of the gray commissure as a bridge of gray matter that connects the left and right sides of gray matter. So if we have any crossing over of the nerves or signals, this is crossing over oftentimes will occur within our gray commissure. So those are the main regions of the gray matter of the spinal cord. Let's focus on the white matter. The white matter is broken up into white columns, and individual columns are referred to as furunculus, or furunculi for plural. And this is all just going to be directional. So we have anterior furunculi in red. We'll use some yellow here for the posterior furunculi on the left and right. And then in green, I'll highlight the two lateral furunculi. So as we look at our directional terminology, if we can give good, proper frame of reference, we are able to better identify what we're looking at. Speaking of frame of reference, it's important to look at these indentations that are here in the white matter. These indentations have some special names. We have the posterior median sulcus and the anterior median fissure. And in particular, when we look at photomicrographs to identify these structures, it's important to find the anterior median fissure. The fissure is going to be a deeper indentation. The sulcus is going to be a shallower indentation. And from this view in particular, it's hard to see, but I want you to think of this shallow indent that I'm indenting or highlighting in red as the posterior sulcus. And then I want you to think of this groove right there in green as the anterior median fissure. And we'll pull up some photomicrographs to make that a little bit easier for you to identify. So on our lab objective sheet, we have our histology guide links. And here we have a cross-section of a spinal cord. And I am not the biggest fan of this image, so let's go back to our lab objective sheet and just pull up the digital histology links. What I want is, there we go. I want that nice clear distinction. So here we can see the gray matter is stained distinctly different from the white matter. The white matter has more blue pigmentation in this image. So we're looking at this spinal cord cross-section. We can see there's this deeper groove and a shallower groove right here. This deep groove that goes this pronounced line, I'll go ahead and highlight this line right here for us. That is our anterior median fissure. So it's a little bit more pronounced than the posterior median sulcus. So look at that anterior median fissure. That's going to be our frame of reference. Now as we're looking at, once we identify anterior, then we're able to identify the horns with a directional terminology and also the furunculi with our directional terminology. Oh, a little too far here. So let's go back to this image. Here on this image, now that we've looked at the easier image, it's always nice to start off with digital histology and then work our way up to the histology guide site. So here on this image, I did not press the space bar. On this website, if you press the space bar, you alternate between full screen and partial screen. Here we can see the anterior median fissure right there on this photomicrograph. It's a little bit more difficult to see, but this is the more representative of what you would see in an actual cadaver specimen. Mostly that it doesn't have the fantastic staining that we're used to seeing. on most of our images, something like this, where it's very clear between white matter and gray matter. As we look at the meninx, which is the singular for the layers of the membranes that wrap around the spinal cord and brain, the central nervous system, meninges is plural. We have Dura Mater, that is most superficial, Pia Mater, which is the deepest, and Arachnoid Mater, which is in between. In this image here, not that we're asking you to identify the meninx or meninges in a photomicrograph, but it's nice to see what we're looking at here. You can see a darker membrane working its way all the way around this cross-section of the spinal cord. And that thick kind of orangish colored membrane, that is the Dura Mater. And then outside of the Dura Mater, we have the epidural space that I'm highlighting in yellow right here. And then deep to the Dura Mater in green, we have the subdural space. And we can see these spaces in visible body with the cross-sectional view. So here's our cross-sectional view in visible body again. So we have in pink in this model the Dura Mater. So outside of the Dura Mater, we have our epidural space, and then deep we have the subdural space. You've probably heard the term subdural hematoma. That's when we have bleeding on the brain deep to the Dura Mater, but superficial to the Arachnoid Mater and Pia Mater. As we look at the Arachnoid Mater and Pia Mater, I want you to reference your lab manual, particularly I believe it's figure 19.2 in your lab manual. Correct me if I'm wrong. That will show the meninges and the epidural spaces here in visible body, and with our cross sections that we have, it's not clearly visible. Moving on, we have nerve structures. We want to talk about the roots of the nerve. And these are for spinal nerves. So here in visible body, I'm going to go ahead and back up. Let's go to our system views. And we'll just go with an overview of the central nervous system, like we started out this video with. And except this time, we're going to fixate on branches of the spinal nerves. So I've cleaned it up. Let's go ahead and get rid of all of the peripheral nervous system, just to tidy up our view. And let's get rid of that Dura Mater too. It's kind of messy. It makes it hard to know what we're looking at. There, this is a happier view. And now that I've removed the entire peripheral nervous system, I'm going to selectively add back in our intercostal nerves. And the reason I'm selectively adding back in the intercostal nerves, actually let's clean that up a little bit. Let's only add in half of them. There we go. Now we're able to see these roots that we need to know. So as we look at these branches coming out from the spinal cord, I want to point out that we have a bunch of filaments on the anterior and filaments on the posterior of the spinal cord that go in and out. These make up the spinal nerve roots. In particular, we have the posterior root and the anterior root. Hey, the names make sense. And they kind of look like the roots of a plant. As we're looking at these roots, let's go down here and focus on this most inferior one, just so that we have a little bit cleaner of a view here with invisible body. So we have the posterior roots, we have the posterior root ganglion, and here we have the anterior root. These anterior and posterior roots will merge together to form the anterior ramus, which is kind of just a way of saying a branching of the nerve. And then the anterior ramus is going to have another branch point coming off of it. I'm going to go ahead and zoom out and rotate our model around so we can better see what we're looking at. Right here, there's another pathway coming off. This pathway here is the posterior ramus. So we have our anterior ramus, and here we have our posterior ramus. Oftentimes, or sometimes still referred to as dorsal and ventral, just because of that quadruped terminology that's been embedded in the literature. So those are the major nerve structures. This is page 2 of your lab objective sheet, and it was an overview of the spinal cord anatomy and generic nerve structures. If you have any questions or comments, please feel free to post them in our class discussion board or to shoot me an email. And as always, happy studies.